Welcome to this lecture on animal biotechnology. Today, we are going to explore three of the most powerful tools in genetic engineering: bacterial, yeast, and mammalian artificial chromosomes. If you have ever wondered how scientists manage to sequence the entire human genome, or how we are developing permanent gene therapies for devastating diseases, you are in the right place. We will build our understanding from the ground up, looking at the biology and the mathematics behind these incredible genetic cargo ships. Let us dive in. To begin our journey, imagine trying to move an entire library using only a small bicycle basket. It would take millions of trips. Standard plasmids are like those tiny baskets. When scientists needed to map massive animal genomes, they invented bacterial artificial chromosomes, or BACs, which act like heavy-duty cargo ships. Engineered from the naturally occurring F plasmid in bacteria, BACs have strict replication control, meaning they remain incredibly stable without unwanted mutations. By stripping the plasmid to its bare essentials, scientists created enough room to carry massive DNA inserts, ranging from 100 to 300 kilobases. Now that we understand what a BAC is, let us look at the mathematics of genomic libraries. When scientists chop up a genome, they need to ensure every single piece is represented in their bacterial clones. The Clark carbon formula calculates exactly how many clones are required. In this equation, N is the total number of clones we need to grow. P is our desired probability of finding the target gene, usually set to 99%. Finally, F is the fractional size of the genome carried by a single vector. As the vector's capacity increases, F gets larger, which mathematically drives down the total number of clones required. Before we move on, let us see this formula in action with a concrete example. Suppose a geneticist is studying a mammalian genome containing 3 billion base pairs. They want to be 99% confident that every single gene is represented in their library. To do this, they use a BAC vector that carries an average DNA insert of 150,000 base pairs. Our goal is to calculate exactly how many individual BAC clones the scientist must generate to achieve this 99% probability. We will solve this step by step. Let us begin solving the problem by finding our fraction, F. We need to determine what portion of the total genome fits into a single BAC vector. We do this by dividing the insert size, which is 150,000 base pairs, by the total genome size of 3 billion base pairs. The result is 5 times 10 to the power of -5. This tells us that each individual BAC clone carries a very tiny, yet highly significant fraction of the entire mammalian genome. This value will now serve as the input for the denominator of our main formula. Now that we have our fraction, we can plug it into the Clark carbon equation, along with our 99% probability. The numerator becomes the natural logarithm of 0.01, representing the 1% chance of missing the gene. The denominator uses our calculated fraction. By evaluating these logarithms, we find that the scientist must generate and screen approximately 92,105 individual BAC clones. While this sounds large, it is a massive triumph. If they used a standard plasmid, they would need over 2.7 million clones. Having proven the mathematical efficiency of BACs, let us look at their physical structure. On screen is a schematic map of a bacterial artificial chromosome. Notice the circular DNA structure. The key components include the origin of replication, labeled ORI-S, and the Rep-E gene, which together control the strict copy number. You can also see the par A and par B genes, which ensure the plasmid is stably partitioned during bacterial cell division. Finally, the large gray region represents the massive insert capacity of 100 to 300 kilobases, where the foreign animal DNA is spliced in at the multiple cloning site. While BACs are fantastic, they operate inside simple bacteria. Animal genes often contain complex features like introns that bacteria simply cannot process. This brings us to yeast artificial chromosomes, or YACs. YACs are designed to trick a eukaryotic yeast cell into thinking a massive piece of foreign DNA is its own natural chromosome. Unlike circular bacterial plasmids, a YK is linear. It contains a centromere for cell division, telomeres to protect the ends, and an autonomously replicating sequence. Because it operates in a eukaryotic environment, a YK can hold mind-boggling amounts of DNA, up to 2 megabases. To understand the sheer power of a YAC, we return to the Clark-Carbon formula. The equation remains exactly the same as before, but the input variables change dramatically. Because a YAC can carry up to 10 times more DNA than a BAC, the value of our fractional size, f, becomes significantly larger. Mathematically, as f increases, the term 1 minus f becomes smaller, making its natural logarithm a larger negative number. Dividing by this larger negative number results in a drastically smaller value for n. This showcases how biotechnology scales with better tools. Let us see exactly how much time and effort the YAC vector saves compared to the BAC vector. We return to our geneticist studying the same 3 billion base pair mammalian genome, still wanting a 99% confidence level. However, this time, they use a yeast artificial chromosome, optimized to carry a massive DNA insert of 1 million base pairs, or 1 megabase. We need to calculate the new required number of clones and compare it to our previous result of 92,000 clones to physically interpret the advantage of using a Y. Just like before, our first step is to calculate the new fractional size of the genome carried by a single vector. We divide the YAC's massive insert size of 1 million base pairs by the total genome size of 3 billion base pairs. The resulting fraction is 3.33 times 10 to the power of -4. Notice that this fraction is nearly an order of magnitude larger than the fraction we calculated for the BAC. Each individual yeast cell is now carrying a much larger, more comprehensive chunk of the animal's genetic blueprint. We now plug our 99% probability and our new, larger fraction into the formula. The numerator remains the natural logarithm of 0.01. The denominator is now the natural logarithm of 1 minus 0.000333. The final calculation reveals that the scientist only needs to screen approximately 13,829 YAC clones. Compared to the BAC, using a YAC reduces the laboratory workload by nearly 85%. This perfectly illustrates why YACs were the vector of choice for mapping the largest eukaryotic genomes. Having seen the mathematical proof of their efficiency, let us examine the structure of a YAC. On screen is a diagram of a yeast artificial chromosome. Notice that unlike the circular BAC, the YAC is a linear molecule, perfectly mimicking a natural eukaryotic chromosome. At both ends, you can see the telomeres, labeled TEL, which protect the DNA from degrading. In the center is the centromere, labeled CEN, ensuring proper separation during cell division. The massive purple region represents the large insert DNA, capable of holding up to 2,000 kilobases of complex animal genes. Now that we have explored cloning vectors, we move to the ultimate vehicles for gene therapy: mammalian artificial chromosomes, or MACs. Traditional gene delivery methods, like viruses, aggressively insert foreign DNA directly into a patient's existing chromosomes, which can accidentally cause cancer. MACs solve this by acting like an independent guest house. They're synthetically constructed microchromosomes that function autonomously inside human cells. Because they are episomal, they remain physically separate from the host's natural genome and never integrate into it. Furthermore, being essentially empty scaffolding, there is theoretically no upper limit to the size of the therapeutic DNA they can carry. To understand the physical advantage of a MAC over a standard plasmid in a mammalian cell, we must look at the mathematics of gene expression decay. When standard plasmids are injected, they do not replicate and are slowly degraded by cellular enzymes. This loss is modeled using a first-order exponential decay formula. Here, C of t represents the concentration of the vector remaining at a specific time t. C0 is the initial concentration. The variable k is the decay constant, representing how quickly the vector is destroyed. For a MAC, this decay constant is effectively 0. Let us apply this decay formula to a real-world medical scenario. A researcher administers an initial dose of 1,000 units of a therapeutic gene to a mammalian tissue culture. If they use a standard plasmid, the vector degrades with a decay constant of 0.1 per day. However, if they use a mammalian artificial chromosome, the vector is perfectly stable, meaning its decay constant is exactly 0 per day. We need to calculate and compare the amount of therapeutic DNA remaining in the cells after 30 days for both vector types to prove why MACs are superior. We begin by setting up the equation for the standard plasmid. We plug in our initial concentration of 1,000 units, our decay constant of 0.1, and our time of 30 days. Multiplying the decay constant by 30 gives an exponent of -3. Evaluating the exponential term, we find that only about 5% of the original plasmid remains. Multiplying this by the initial 1,000 units leaves us with approximately 50 units of therapeutic DNA. This means the gene therapy has essentially failed after just 1 month due to biological degradation. Now we evaluate the MAC vector using the exact same formula. Because the MAC is recognized as a natural chromosome, it replicates perfectly and has a decay constant of 0. Multiplying 0 by 30 days gives an exponent of 0. Since any number raised to the power of 0 is exactly 1, the initial concentration is simply multiplied by 1. The final answer is 1,000 units. This means 100% of the therapeutic DNA is still present and functioning perfectly after 30 days. This mathematical proof illustrates why MACs are the holy grail of permanent gene therapy. To visualize this permanent stability, let us look at a diagram of a cell containing a mammalian artificial chromosome. Inside the nucleus, you can see the host's natural chromosomes drawn in gray. Alongside them is the MAC, highlighted in blue. Notice that the MAC is episomal. It exists completely separate from the host genome, ensuring there is no dangerous integration or disruption of healthy genes. You can also see the mammalian centromere on the MAC, which guarantees that every time the cell divides, the therapeutic gene is stably copied and passed on to the new cells. We have covered a tremendous amount of ground today. So let us summarize our key takeaways. First, bacterial artificial chromosomes are the heavy-duty workhorses of cloning, using the F plasmid to stably carry large DNA fragments and drastically reduce genomic library sizes. Second, yeast artificial chromosomes bridge the gap to eukaryotes, providing a linear structure that can hold up to 2 megabases of complex animal genes. Finally, mammalian artificial chromosomes represent the pinnacle of vector engineering, offering an infinitely large, perfectly stable, and incredibly safe episomal vehicle for permanent gene therapies. Thank you for joining this lecture.